Hi everybody! In this video, we are going to be talking about enzymes. And as per usual, for all of these videos, we're going to start by why you should care, what the big deal is. So for that, in terms of enzymes, we're going to talk about what enzymes are and what they do, which ties into why they're so important. So this is our introduction. So enzymes are a kind of protein. They're a super important class of proteins. So you can think of proteins as being the big umbrella category, and enzymes fall as a smaller category within that umbrella. And their function is to speed up, and the word that we use for this in biology is we can say that they catalyze chemical reactions. And most often, when we're thinking about what kind of chemical reactions these enzymes are going to be speeding up or catalyzing, we're thinking about metabolic reactions. And metabolic reactions, or metabolism in general, is just referring to all of the sum or the totality of all the chemical reactions within an organism. And so all of the chemical reactions that need to take place for a process like cellular respiration to occur, that would fall under the category of metabolism. Any chemical reaction that needs to take place for photosynthesis to occur, also metabolism. Any chemical reaction that needs to occur in order to build up some sort of structural component within a cell, so maybe building up like the cytoskeleton, that's going to be falling under the category of metabolism as well. So any chemical reaction that's taking place within an organism, that's a metabolic reaction. And pretty much every metabolic reaction within a cell is going to require or use an enzyme to speed it up. So from this, you can hopefully see that enzymes are playing a role in so many chemical reactions that are taking place in our cells. And these reactions wouldn't be possible unless enzymes were there to make them go a little faster. Here are a couple more points about enzymes and the reactions that they catalyze. So enzymes are not destroyed by the reaction. They're not used up in any way. So the same exact enzyme can catalyze multiple chemical reactions without being depleted or destroyed in any way. Each enzyme is also specific to a particular reaction. So you can think of this if you've ever worked with screwdrivers or something like that before. You know that there's a flathead screwdriver and a Phillips screwdriver. There's hex screwdrivers, and so it's like that, but times thousands, because each enzyme is specific to one particular chemical reaction. So if you think about all of the thousands of reactions that are taking place within a cell, each one requires its own specialized enzyme. Okay, so that's our overview of what enzymes do. And here, we're just going to go over a little bit of vocabulary that we'll be using throughout the rest of this when we're talking about enzymes and their different components and parts. So one word that we're going to be using is this idea of a substrate. And a substrate in the context of enzymes is just a molecule that's going to be undergoing a reaction. It's the thing that the enzyme is working on. If you're thinking about it in terms of chemistry, it's the reactant, the starting thing, basically. So here in this diagram, you can see that the substrate is these two green shapes that are stuck together. And the next step involves the active site. So the active site is part of the enzyme itself. It's a specific region on the enzyme. So it's a space on the enzyme where the substrate is going to bind. So the enzyme is a bigger molecule, and so there's going to be a certain region of it, a specific part, where the substrate is going to fit in or sort of attach. And the shape of the substrate is going to, or the shape of the active site is going to change a little bit once the substrate binds. We call this induced fit. So you can think of the active site as sort of being like, it's almost the right shape for the substrate, but then once the substrate actually comes and sits down, it changes its shape a little to be more perfectly fitting to that substrate. This is like, I don't know, like leggings or something. They're not quite exactly the right shape for your legs, but once you put them on, they change their shape to be the exact right conformation. And then finally, once the enzyme and once the active site on the enzyme and the substrate are together, then the reaction can actually take place. And the substrate is going to undergo some sort of chemical reaction, some sort of change. And once that happens, it's going to change from the substrate to the products. So the products are just the molecules that are as the result or the product, or yeah, the product of the reaction itself. So the ending place for these molecules. So it's going to start as the substrate, end up as products, and that change is going to happen or come about because of the substrate binding to the active site. And then I've just got a little bit of a note down here that I just covered up by making this menu come up. So the active site shape and its chemistry. So thinking about like what kind of amino acids are present within that active site are very specialized to the substrate that's supposed to bind. So I was saying in the last slide that there are thousands of enzymes, and each one is specific to its own particular reaction. So if you're thinking about each reaction as having a unique substrate or a unique starting molecule, that means that each enzyme's active site is specialized for its own unique substrate or starting molecule. And then once that substrate binds, there's some sort of physical or chemical stress that's encouraging bonds to be broken or formed, so encouraging that reaction to take place. And this is showing the same thing in just a little bit more of a realistic model. So here we're showing the enzyme in blue, and this is called a ribbon model, and it kind of shows the backbone of the enzyme. And then if you fill it in with actual sort of a space filling model where the molecules are filled in with these bubble looking structures, the active site is this pocket right here. So this is where the substrate's going to come in and it'll fit right into this little pocket, and that's shown here in yellow. And then these arrows are trying to show that idea that the active site is going to change its shape a little bit once the substrate binds in order for the chemical reaction to take place. So let's look at an example. So this is an example of an enzyme called lactase. And a lot of enzymes end with this ending, this ACE ending. So if you see a molecule and it's got an ACE at the end, it's a pretty good guess that that molecule is an enzyme. So lactase is an enzyme that breaks down lactose, which maybe that sounds familiar to you. It's one of the disaccharides we talked about when we were thinking about carbohydrates. And it's composed of two monosaccharides, glucose and galactose. So glucose and galactose are monomers that can be put together to make this disaccharide, lactose. And so this enzyme is going to be breaking down lactose, and you can see that right here, the enzyme lactase and this molecule lactose right here. And so lactose is going to be the substrate because it's sort of the starting piece that we're going to be fitting into the active site. So lactose is going to fit right in to the lactase active site, and it's going to be broken up into its two component parts. So you can see after it's bound, when the products come out, they're not connected anymore. They're separate. So we have one single monomer of galactose and one single monomer of glucose. And so those two molecules, glucose and galactose, are the products. So we've got the substrate, lactose, and the products, glucose and galactose. Um, and then, like I was saying, that active site is going to be very specific for the molecule that it needs to be starting out with, very specific for its substrate. So you can see here how this active site for lactase, this is very stylized, obviously, but it's got the exact right shape for the lactose molecule to fit in. So if another molecule comes along, like this is showing sucrose, which has two glucose molecules, it's not going to fit into the active site for lactase because lactase isn't trying to break down sucrose. It's only trying to break down lactose. Um, and then an interesting fact, most mammals are only produce lactase, this enzyme to break down lactose, during infancy when they're still nursing. And so our ability, if you're lactose tolerant, that's actually it came about from a mutation, and this mutation has arisen multiple times in multiple places throughout the world. But the default condition is to be lactose intolerant. Okay, so this is categorizing. We were talking about metabolism in that first slide, and this idea that metabolism encompasses all of the chemical reactions that are occurring in an organism. And so this slide is breaking down this idea of metabolism into two of the main categories that we can think of when we're thinking about metabolic reactions. And those two categories broadly have to do with whether we're building a molecule up or whether we're breaking a molecule down. So synthesis is a type of metabolic reaction where we're building things up. We're adding things together. If you think about the dehydration reaction that we talked about in our carbohydrate unit, that's an example of a synthesis reaction because we're building things together or we're making things bigger. And then the opposite is a decomposition reaction. So this is a reaction where we're breaking things down or making things smaller. So if you remember the hydrolysis reaction that we talked about in the carbohydrate unit, that would be an example of a decomposition reaction. And two other words that we can associate with these are anabolic. So synthesis reactions are also said to be anabolic, and decomposition reactions are said to be catabolic. So if you've ever heard of like bodybuilders using anabolic steroids, this idea of anabolic, it has to do with like bulking up or getting bigger. And then the opposite is catabolic, which goes along with this idea of decomposition. Okay, so this slide is thinking about how we can regulate whether enzymes are active or not. So thinking about how we can turn them on and turn them off. And the main way that we do this has to do with this idea of an inhibitor. And an inhibitor is kind of exactly what it sounds like. It's a molecule that is going to bind to an enzyme and prevent it from working. So it's going to sort of turn the enzyme off. And there are two main ways that this can happen. So this inhibitor can bind either at the active site itself. So if you're thinking about this enzyme, we're saying that this pocket right here on the top is the active site, and you can see that it sort of looks like it'll fit this substrate. Let's imagine this is a perfect little spot for this substrate to fit. So the active site is right here, and if an inhibitor just sits right down inside of that active site pocket, then you can see that it's blocking access. So the substrate can't bind because its like seat is being taken up by this inhibitor. So we call this competitive inhibition because they're directly competing for access to that active site. It's almost like if you've ever played musical chairs, you know how there's one less chair. So it's like the active site's the chair, and both the substrate and the competitive inhibitor are trying to sit down on it. So if the inhibitor wins, then the substrate can't get that chair, can't get down to the active site. So that's one way that it can happen, and we call that competitive inhibition. The other way that it can happen is if the inhibitor binds somewhere else. So here you can see that the inhibitor is not binding at the active site. The active site's still totally empty, but by binding to this other region, and we call this an allosteric region, it's going to change the shape of the active site itself so that now the substrate doesn't fit there anymore. So the substrate can't bind even though the active site's empty. So in this, we call it non-competitive inhibition because the inhibitor is not actually competing with the substrate for the active site. It's going somewhere totally different. But by binding to that other place, we're changing the shape of the active site so the end result is the same, that the substrate can't bind. And so in both of these cases, the substrate's not getting to the active site, and so this reaction, whatever reaction the substrate is supposed to be undergoing, is not going to happen. So that's in effect turning the enzyme off and not allowing that reaction to go forward. Okay, so the last few slides are all going to have to do with this central question, and that is, what affects the rate of an enzyme catalyzed reaction? And so when we're thinking about this idea of rate really has to do with speed. How quickly is our enzyme able to carry out its function? How quickly can the enzyme do what it's supposed to be doing? So if you think about the example that we had earlier with lactase enzyme, this question would be, how quickly can lactase be breaking down lactose? And this could be the difference between whether you are able to only have maybe like a splash of milk in your coffee, and that's just a little bit of lactose. So even if your enzyme lactase is moving really slowly, like maybe it's okay. But if you chug a glass of milk, then we're going to need lactase to be going really quickly in order for all of that lactose that you just drank to get processed. So when we're thinking about this question, there are two big components that we should be thinking about. And those two components are how often the enzyme and the substrate are colliding with one another, So this has to do with the idea that in order for a substrate to undergo whatever change it needs to go undergo, in order for that reaction to take place, we need to have the enzyme and the substrate to actually meet each other physically. They have to bump into one another because the substrate has to fit into that active site. And if they never touch, then that's never going to happen. So, how often they're going to collide, the enzyme and the substrate. And then, secondly, whether the enzyme and the substrate can fit together properly. So, I already talked about how the active site is the site, the shape of that active site is crucial for the substrate to fit into it and only the substrate to fit into it. So, whether they fit together in the right way, that's going to be another factor to consider. And so, we're going to think about three different major types of factors that can affect reaction rate. And those three factors are whether there are regulators present. And by regulators, I mean inhibitors. And we also have activators, which are the opposite of inhibitors. And then there are also things that have to do with concentrations. So we could think about whether there's a lot of substrate or just a little bit of substrate, whether there's a lot of enzyme or just a little bit of enzyme. Substrate and enzyme concentration will have an effect on the reaction rate. And then finally, the external conditions that are surrounding whatever this reaction is. So things like temperature and pH will also have an effect. So we'll look at these one at a time. So first, let's think about this idea of regulators. So we've already kind of talked about this with the idea of inhibitors. So hopefully this is not a surprise. Inhibitors are going to reduce the reaction rate because, like we were talking about, they're going to prevent a proper interaction between the enzyme and the substrate. So remember we said so this is showing an enzyme and it's showing an active site right here. And so our substrate should be coming to sit down right in this active site. But we talked about how sometimes you get an inhibitor that can bind to the enzyme and cause that active site to change shape so that the substrate doesn't fit. And if you remember from our inhibition slide, you should recognize this as being non-competitive inhibition. Since we're not competing for the active site itself, we're binding somewhere else and distorting the shape of this active site. So it's preventing proper interaction between enzyme and substrate, which is going to reduce the reaction rate. And then conversely, activators can increase the reaction rate, so encourage proper interaction between enzyme and substrate. And this can happen in a couple different ways. Some enzymes are by default, the active site isn't shaped right, and it isn't until something comes in and binds to an activator site, and then that's going to change the active site to be like the proper shape for the substrate to come fit in. Some activators block this inhibitor binding spot so that the inhibitor can't come in and reduce the rate. So activators can do a couple different things, but the main point is that they're increasing the reaction rate. Okay, so that's regulators. Oops, our next factor has to do with different kinds of concentrations. So the two main players when we're thinking about the reaction rate are the enzyme itself and then the substrate or the molecule that we're starting with. And if you increase the concentration of either one of these, you're going to increase the rate at which you're going to increase the rate that the reaction is happening at. And this is because when you have more molecules, be it enzyme or substrate, they're going to collide more often, right? That's just like if you think about having more people in a room running around, they're going to bump into each other more often the more people you have, right? So this is looking at we've got these little purple molecules are the substrate, and these pinkish bean-shaped molecules are the enzyme. And so here you can see if you have just a little, you always have three enzymes that's staying constant. But if you only have a little bit of substrate, they're not going to bind to, or they're not going to bump into each other very often. And so you can see that the only one of those three enzymes has a substrate attached to it, and the reaction rate's kind of low. But if you increase the number of substrate molecules you have, then there's more of a chance for them to bump into these enzymes. And so the reaction rate's going to be increasing as you increase the substrate concentration. And then the only tricky thing is that eventually you can get to a point of where you're not getting any extra benefit from having more molecules. So we call this point saturation. So this idea is like we only have three of these enzymes in this picture. And so even if we keep adding more and more and more substrate, all of these three enzymes are full and they're already going as fast as they can. And so adding more enzymes or adding more substrate is not going to help because they're already bumping into each other at the maximum capacity rate. So eventually you get to this point called saturation where the rate doesn't increase anymore, it just sort of levels off. And that's because we're already going as quick as we can. So increasing the number of substrate or increasing the concentration of substrate, increasing the concentration of enzyme, both of those things will give you this increased reaction rate up to the point of saturation. And then second to last, we're going to talk about temperature and pH separately. So temperature, higher temperature or adding heat to reactions will generally increase the reaction rate. And this is because adding heat to a system is going to make the molecules in that system move faster. And this is going to make them collide more often. So you can think about, I don't know, a bunch of people walking around in a gym versus a bunch of people like running as fast as they can through a gym. There are going to be more collisions when people are running, right? So all of these extra collisions, which we were talking about, the enzyme and the substrate need to collide. So these extra collisions, because things are moving faster, will mean that the reaction rate is going to be higher. So here you can see that happening on this graph. So as you get hotter and hotter and hotter, the rate of the reaction is going up and up and up. But you can also see that it sort of levels off right here. And that's because at a certain point, it actually gets to be too hot. And what can happen is that you'll denature the enzyme. And if you remember, denaturing a protein means that you lose the proper shape for the protein. It becomes sort of unfolded. And enzymes are proteins, so they will denature as well if it gets too hot. And once the protein is denatured, since it loses its shape, for an enzyme, what that means is that the enzyme is losing its shape and the active site's not going to be structured correctly anymore. So it's not going to be able to do its job. So that way you can see that the rate of reaction is just going to take a steep drop off once the temperature gets too hot. And here, this second bullet point is just referring to the fact that each enzyme is going to have an optimal temperature where its reaction rate is highest. So that's just referring to this peak on this temperature graph. There's a certain temperature where you're going to get the most bang for your buck, where we've increased the temperature enough that reactions are happening more quickly, but we haven't reached the point of denaturation yet. And this is going to be a little bit different for different kinds of enzymes and especially different kinds of organisms. So if you think about bacteria that are living in like near heat vents or something like that, their enzymes are going to have a much higher optimal temperature than, for example, what we would have. So different organisms and different enzymes can have different optimal temperatures. And then this is the last slide. So pH. In the same way that each enzyme has an optimal temperature, each enzyme also has an optimal pH where its reaction rate is the highest. So here we're looking at the optimal temperature for 3 different enzymes. Pepsin is a digestive enzyme in your stomach, so you can see it as optimal at a really low pH. Salivary amylase is like a saliva enzyme, so that's a pretty neutral pH where it's happiest. And then alkaline phosphatase is an enzyme that's really common in bile ducts and your intestine. And bile is an alkaline substance, so it works best in these alkaline pHs. And in the same way that high temperatures can denature a protein, if the pH isn't quite right, if it's too high or too low, that can cause the proteins to be natured as well. So this enzyme, if it gets denatured with the wrong pH, again, its active site won't be shaped correctly and the reaction rate will decrease. Okay, so those are, I think, yes, all of our slides. And yeah, I hope that this made sense. Enzymes can be a little bit of a weird concept to think about, so we'll practice more about this in class, and I'll see you guys soon.